Hello, everyone, and welcome to another seminar from the RNA Collaborative Seminar Series. Just a reminder, this series is sponsored by the RNA Society, and today's seminar is being hosted by the NCI RNA Biology Initiative. I'm very excited about our two presenters. We have two outstanding talks by tenure-track investigators at NCI and NIA here at NIH. Before I present them, I just want to remind you that if you have questions, you can type them in the Q&A box, not in the chat box, during the seminar or after the seminar, and I will relay them to the speaker at the end of their talk. And if they don't have time to answer all the questions live, they will then go over the chat and reply to them individually. And so with that, I would like to introduce the first speaker of today, Thomas Gonatopoulos-Pournatzis. He's a tenure track at NCI, and he's going to tell us about how his group is using genome-wide screens to understand which exons are essential in cells. Take it away, Thomas.
Thanks. Thank you, Joana. I don't know if you can see my slides.
Perfectly.
Okay, great. Thank you, and thank you everyone for joining this RNA collaborative seminar. So today we'll present you some recent progress of my group directed at the systematic identification of the exons in the human genome that underlie critical cellular phenotypes. So my research group is focusing on studying alternative splicing, which, as you all know, is a highly regulated mechanism by which a single gene, through the selection of different splice sites, can generate multiple mRNA and frequently protein isoforms with the potential to diversify gene function and amplify the coding potential of the genome. High-throughput sequencing studies of transcriptomes originating from various species and cell types have revealed that alternative splicing is a widespread phenomenon, especially in human cells, in humans, where it affects almost all multi-exon protein coding genes. And such studies have also shown that spliced isoforms are frequently very precisely regulated and controlled in different cell and tissue types, developmental stages, or dynamically regulated upon changing environmental conditions, implying functionality. Thus, it is not surprising that misregulation of alternative splicing has been implicated in a myriad of diseases and disorders, including autism, genetic syndrome such as progeria, but most notably cancer. Indeed, the transcriptomic analysis have highlighted pervasive splicing alterations of splicing patterns in tumors, alterations that provide plasticity to cancer cells and impact almost all hallmark features of cancer, such as increased proliferation or resistance to apoptotic signaling, for example. These alterations result either from mutations or dysregulated expression of key splicing factors. For example, increased expression of the splicing regulator SRSF1 in breast, lung, and other cancer types can drive a protemerogenic splicing program, and also recurrent hotspot mutations in splicing factors such as SF3B1 and U2F1 are frequently observed in certain leukemias, among other cancer types, again resulting in widespread splicing alterations. Despite the significant progress, though, in cataloging thousands of alternative splicing events in normal physiology and disease state, the functional significance for most spliced isoforms remains elusive. Therefore, a key question in biomedical research is to determine which alternative exons in the human genome harbor functional importance. And of course, this challenge is all the more important for cancer cells characterized by splicing factor mutations, for which the most consequential downstream target remain controversial. So today we'll present you our efforts to start addressing this challenge by developing, optimizing, and applying exon resolution functional genomic strategies to systematically identify exons in the human genome that impact critical phenotypes such as cell fitness and proliferation, a powerful and quantitative phenotype with cancer elements, but also phenotypes measured by transcriptomic signatures, something that I will focus on the last part of my talk. So to address these challenges, we are using Chimera, a tool we developed when I was a postdoc in Ben Blenco's lab at the University of Toronto, together with Jason Moffat's group and Mike Larager, who is currently a PI at NCI. Chimera is based on the expression of two orthologous Cas protein, Cas9 and Cas12a, expressing the same cell together with a hybrid guide RNA that is engineered by the fusion of the Cas9 and Cas12a guides expressed from a single UC6 promoter, ensuring equal expression of the two guides, which is critical for appropriate editing. However, this guide is initially expressed as a single hybrid guide RNA, which is actually processed by the intrinsic RNase activity of Cas12a. This is a unique feature of Cas12a compared to Cas9. It can recognize its own scaffold sequence and induce an endonucleolytic cut upstream of its own direct repeat, liberating the two guide components that can then form complex with the corresponding nuclease for combinatorial editing. And we can program the system by designing the appropriate spacer sequences so that the two nucleases can target flanking intronic sites of exons of interest for deleting them in a massively parallel manner through designing hybrid guide RNA libraries. However, achieving exon deletion is far more challenging than conventional gene knockout screens, since it is based on the timely coordinated and equal efficacious editing of the two Cas proteins. So when we started at NCI, we invested a substantial amount of time to improve Cas12a editing activity, which was a limiting factor in the initial iteration of Chimera, by testing various variants, modifications, and other aspects of the system. And indeed, we now have in our hands an enhanced Chimera version, which is described in our recent publications, that we have used for all the data I will show you today. So with this enhanced Chimera in hand, Mei-Seng Shao, a postdoc in my group, went on and designed an elaborate hybrid guide RNA library consisting of more than 300,000 hybrid guide RNAs that allows to target more than 12,000 exons in the human genome for deletion in order to identify which of those can affect cell fitness. So with this library, we mostly focused on frame-preserving exons, or else exons whose deletion is expected to generate different protein variants, rather than inactivate the corresponding gene. So in other words, with this library, our goal was to interrogate the exon-encoded protein regions that can affect cell fitness. We also targeted frame-disruptive exons as controls, and the exons we targeted were both alternative and constitutive, residing in both essential and non-essential genes. So after we in silico designed the library, we cloned it into lentiviral vectors, which we used to generate virus and transduce cells expressing Cas9 and Cas12a at a low multiplicity of infection, such that each cell is infected or expressing a single hybrid guide RNA. And we perform these pool screens, meaning that we generate a pool of 300,000 different mutations, or else cells expressing 300 different hybrid guide RNAs, with a coverage of 200 to 300 cells for each hybrid guide RNA, meaning that every time we passage our screens, we passage along 60 to 90 million cells for each of the 3 replicates. And we extract genomic DNA from the very beginning and the end time point of the screens in order to amplify the hybrid guide RNA expression cassettes, which we sequence them and determine which are the guides that are depleted or enriched from the population over the course of 2 to 3 weeks that it takes to do the screens. And this information can tell us which are the exons that can promote or suppress cell fitness respectively, with the assumption, of course, being here that that guides that target exons essential for cell fitness, they will be readily depleted from the population over time. So we performed the screens initially into HAP1 leukemic cells and RP1, it's retina epithelial immortalized cells, identifying more than 2,000 frame-preserving exons that can affect fitness, with a significant overlap between the two cell lines. And our screens revealed both fitness promoting, mostly exons, but also some fitness suppressing. So fitness promoting, we call the exons that when skipped, the cells have a self-fitness defect, while fitness suppressing, we call those that when removed, the cells can actually grow better. Also, I would like to point out that the phenotypes we observe are due to exon deletion because when we analyze control hybrid guide RNAs that are based on single intronic cuts, while the second guide targets an unrelated intergenic region, we fail to detect a significant number of hits, again suggesting that these phenotypes are due to exon deletions and inducing intronic mutations. So we could validate over 40 of these exons with focused exon deletion assays coupled to proliferation with very good validation rates. However, one concern we had is the extent to which some of the phenotypes we observed are due to not due to deleting the exon, but due to the multiple double-stranded breaks inducing the genome. So we wanted to also validate the data by applying an orthogonal approach, and we reasoned that base editors will be ideal for this. So the idea here is that instead of deleting exons, we can recruit base editors to splice sites so that could mutate either the adenine or cytidines of the splice sites, as indicated here, to induce exon skipping. So Ethan Dixon, a former postdoc in the lab, optimized base editors by testing various Cas proteins and deaminases and various variants of them. And he did identify some that result in very efficient splice site mutation and exon skipping, illustrated by this RT-PCR agarose gel monitoring the splicing of two alternative exons in TAF5 or NRF1 genes. I will talk about these exons later in the talk, but for now, I just hope you can appreciate that when we mutate the splice sites, we get efficient, very efficient exon skipping indicated by the lower molecular weight band here. So with this tool in hand, Ethan and Mason designed a library targeting an overlapping set of exons with the exons we targeted by the exon deletion library. And we're very happy to detect a very strongly significant overlap of exons to affect cell fitness by both strategies and an overall positive correlation of log-fold change values, despite using orthogonal perturbation systems and very different guide RNA libraries. So with this extra confidence about the quality of our screens, we went on to analyze our rich dataset. And an initial interesting observation we had is that more than 20% of the interrogated frame-preserving alternative cassette exons can affect cell fitness, highlighting that a very high fraction of alternative splicing events can regulate protein activity, playing critical regulatory roles and affecting measurable cellular phenotypes. Another interesting observation was that the fitness promoting and suppressing exons have largely distinct characteristics. Just to remind you, fitness promoting, we call the exons that when skipped, the cells have a self-fitness defect, while fitness suppressing are those that when not included, the cells can grow better. What we observe is that the fitness promoting exons tend to reside in essential and highly expressed genes. They have high inclusion levels and they're more conserved. And all these are in striking contrast to fitness suppressing exons, which in turn they tend to overlap disorder regions and clinical mutations. However, these two groups of exons share some commonalities. They both tend to overlap protein domains and protein interaction interfaces, highlighting that a mechanism by which splicing can affect fitness is by controlling protein-protein interaction networks. So all these observations were very interesting, but one of the major motivations for us to perform these screens was also to prioritize individual exons for focused mechanistic studies in order to better understand how alternative splicing can control biological processes of interest. And as a lab, we're generally interested in gene expression and regulation. So Arun Prasad Damodaran, a postdoc in the group, took advantage of this rich data set and focused in a few interesting alternative exons, starting by the alternative exon 8 in the transcription factor TAF5, an exon that was a very strong hit by both the exon deletion and base editor screens. So TAF5 is a component of the TF2D general transcription initiation factor, consisting of 13 TAF proteins, 13 subunits, and the TAT-abiding protein, and ultimately recruits the RNA pol2 to the promoters of eukaryotic genes to drive gene expression. So Arun initially took an unbiased approach and purified full-length and delta exon Ta5 complexes from 293 cells and subjected them to mass spectrometry analysis. So with the full-length Ta5, we can purify pretty much the whole TF2D holoenzyme. However, when this single exon is removed, we fail to detect even a single peptide corresponding to another TF2D subunit. And that was something that we could validate by QIP Western, shown here. For example, it's only the full length that associates with TBP and not the isoform lacking exon 8. So Arun then asked, does this exon simply controls the ability of Ta5 to interact with TF2D, or it even controls the overall TF2D complex formation? And to address that, he purified the TATA-binding protein from cells, he immunoprecipitated TBP, and assessed the ability of TBP to interact with TF2D subunits such as TAF1, 10, and 12 as a means to assess TF2D complex formation. So depletion of Ta5 results in reduced association of TBP with TF2D subunits, something that we can rescue by adding back the full-length Ta5 protein, but not the isoform lacking exon 8, suggesting that this alternative exon 8 is critical for TF2D complex assembly. And considering that, we're not really surprised to also find out that this exon is required for TF2D dependent gene expression. So to test that, we perform, again, these isoform rescue experiments where we deplete endogenous Ta5 using RNAi, and we complement by adding back siRNA-resistant isoforms, either the full length or the delta exon 8 isoform, expressed at near endogenous levels. So depletion of Ta5 results in widespread gene expression changes, something that we can largely rescue by adding back the full-length Ta5 protein, but not the isoform lacking exon 8. So this data suggests that Ta5 alternative exon is critical for its transcriptional activity. And we further showed that this exon, by performing chip sequencing experiments, that this exon regulates gene expression by controlling the recruitment of the TATA-binding protein and RNA polymerase II to the promoters of regulated genes. Finally, we also ask how this exon is regulated. And to address that, we perform a small siRNA screen. So Arun knocked down around 60 splicing regulators and monitor the endogenous of this alternative exon inclusion. And among the interrogated factors, we identify SRSF1 as the splicing factor that could have the strongest effect in promoting exon 8 inclusion. Interestingly, we also observe a perfect binding SRSF1 motif within this alternative exon 8. And then these mini-gene experiments, this RT-PCR shown here, show that either by knocking down SRSF1 with 3 independent siRNAs or mutating this motif, both results in strong exon skipping, suggesting that SRSF1 likely binds and promotes exon 8, Ta5 exon 8 inclusion. Furthermore, we have shown that this exon displays increased skipping following genotoxic stress, and taken together, this data highlight an elegant regulatory mechanism by which the alternative splicing of a single exon in a megadalton protein complex such as TF2D can control the overall stoichiometry and activity of this complex in the cells, impacting gene expression. Furthermore, this work validates the success of our high-throughput exon perturbation methodologies for characterizing novel biology. So this work was published a few months ago, and the screens we have conducted and I talk about so far rely on cell fitness as the primary readout. And while cell fitness is a highly quantitative and a straightforward phenotype to implement, it provides limited mechanistic insights. Specifically for each exon perturbation, we obtain a single log-fold change value tied to cell fitness, which is a relatively broad phenotype and may obscure more nuanced functional effects. So to overcome this, we have extended our screens by focusing on information-rich phenotypes offered by single-cell transcriptomics. So with this experiment, each exon deletion yields expression data for thousands of genes, providing high content information. This project is in collaboration with Mike Larager, PI at NCI, and is driven by Vandana Kumari, a postdoc in my group, with very important contributions from Wilfred Guible, a computational scientist at the RNA biology lab. So through extensive optimization series, we have made our Chimera exon deletion platform compatible to 10x genomic single-cell RNA sequencing phenotypic readouts, generating a tool which we call single-cell Chimera-seq. For the interest of time, I will not disclose all the tricks and modifications we have to implement to convert Chimera to a tool-enable exon deletion coupled to single-cell RNA sequencing. But just I want to briefly mention that the single-cell Chimera sequencing allow us to very effectively capture not only mRNA and the Cas9 guides, but also the Cas12a guide RNAs at the single-cell level, while maintaining or even enhancing exon deletion efficiency. So this allows us to link the deletion of individual exons with specific transcriptomic signatures. We have applied single-cell Chimera-seq to delete over 200 alternative cassette exons in the human genome to determine their impact on the transcriptome. And we use multiple hybrid guide RNAs for each section, including various controls, including many intergenic targeting guide RNAs for negative controls. And in total, we profile approximately 200,000 cells, corresponding to more than 500 cells for each deleted exon. We have only very recently acquired this data, and this is very much work in progress. And to analyze those, Vandana is applying Seurat to calculate the perturbation signature of each cell and then cluster cells based on gene expression profiles as shown in these UMA plots. We then perform pseudobulk analysis of cells grouped based on the expression of the same hybrid guide RNA. Or else we group together cells that have the same exon deleted, and we perform differential gene expression comparing cells infected with intergenic control guide RNAs. And this analysis revealed that a substantial number of the targeted exons, close to 40%, or else 87 among the 224 targeted exons, can regulate the expression of numerous genes. One of these, which the cutoff we apply for this analysis was 50 genes. So one of the exons we identified to affect the cellular transcriptome through this experiment is an alternative exon in the transcription factor NRF1. So NRF1 is a transcription factor that can recognize specific motifs at the promoters of the gene it regulates. And as I mentioned before, this pseudobulk analysis of cells transduced with guides aiming to delete NRF1 exon or intergenic controls reveal close to 200 genes affected by this alternative exon. So to validate this data, we follow a similar approach as the one we follow for the TAF5 alternative exon. So Arun did the exact same things. He generated cell lines expressing the two isoforms. Cell lines are resistant to the most efficient siRNA. We could optimize targeting NRF1. And as indicated in this heat map here, when we deplete endogenous NRF1, there are gene expression changes that can be rescued by adding back the full-length NRF1 isoform, but not the isoform lacking this alternative exon, suggesting that this exon is important for NRF1 transcriptional activity, validating our single-cell data. Mechanistically, this exon controls the ability of NRF1 to bind to the promoters of regulated genes. ChIP sequencing of the full-length and delta exon NRF1 isoform shown here indicates that when this exon is removed, NRF1 has a much weaker affinity for binding to the promoters of the gene it regulates. Again, suggesting that this alternative exon can control NRF1 transcriptional activity through controlling its ability to bind to DNA. So I will finish my talk by a simple comparison of the single-cell chimera sequencing data with those we obtained by the focused isoform rescue experiments. So despite that these experiments have been done in different cell lines, in HAP1 cells for the single-cell experiments or flipping 293T cells for the isoform rescue assays, we're very happy to detect a very strong overlap between these two approaches and an overall positive correlation of log-fold change values. However, it is worth considering that the single-cell experiments can be performed in 1 to 2 weeks and allows detecting the impact of hundreds or even thousands of exons in gene expression. On the other hand, the focused isoform rescue experiments take several months and involve optimizing siRNA sequences, cloning and mutating the open reading frames to make them resistant to the siRNA, generating stable cell lines, ensuring equal expression, and so on. And at the very most, we can focus on 1 or 2 exons at the time. Therefore, we believe that the single-cell chimera-seq represents a very powerful methodology for the massively parallel deletion of human exons in order to identify those that affect gene expression with potentially transformative implication in the systematic study of alternative splicing. So to wrap up, what I've shown you here is the development of methodologies allow us to delete exons and that can be integrated with cell fitness or single-cell RNA sequencing phenotypes. A substantial number of the profile exons can affect these phenotypes, highlighting the importance of alternative splicing in protein activity and playing critical regulatory roles. These approaches have the potential to identify critical exons for normal physiology, but also disease-related phenotypes, and we're indeed currently applying these tools to various cancer models with colleagues at NCI. Finally, our program has uncovered novel mechanism of how alternative splicing through controlling protein-protein interactions or protein-nucleic acid interactions can regulate gene expression. And with that, I will finish by thanking my group. First, current and past members. I tried to mention the key players during the talk. Important collaborators such as Mike Larriger, with whom we have co-developed Chimera and single-cell Chimera-seq. Rob Weatherit and Joana Vidigal for helping annotating exons and looking for Cas12a off-target effects. And many other collaborators in the RNA biology department at NCI for generating a very stimulating and supporting environment for RNA research. I'm happy to take any questions.
Thank you, Thomas, for an amazing talk. We have very little time for questions, but anyone, please write your questions in the Q&A, and Thomas will be able to answer them during the next talk. I will take just a few minutes that we have to ask. Chris, can you please write your question in the Q&A? And I will read it out. Okay, I think Chris will write, and maybe you can reply in writing while Manolis is giving it the talk. But in the meantime, I have one question for you, which is, do you know under which circumstances the exon of TAF5 is included or excluded?
So yeah, I mean, we have some data indicating that stress, particularly genotoxic stress, can induce exon skipping. To what extent this is sufficient to affect transcriptomic, the activity of the TF2D complex, we haven't looked, but genotoxic stress results in to some extent to exonate skipping.
Through regulation of SRF1 function.
Yeah, that's also not something we have directly shown, but presumably these two are implicated. Yeah.
Okay, with that, we will move for Manolis' talk. Thank you so much, Thomas. Manolis, our next speaker, is coming to us from the National Institute of Aging, and he will tell us about the regulation of RNA stability and translation in stress response and senescence. Take it away, Manolis.
Okay, I think I'm unmuted. Okay, so okay, thank you very much for the introduction and for the invitation to speak to the RNA seminar series, and I'm very happy to be presenting. So in today's talk, I'll try to summarize quite a bit of work that has been going on in my group over the last few years with respect to trying to understand regulation of RNA stability and translation in stress response and senescence. So being in the National Institute on Aging, we obviously want to understand aging as a process, both in physiology and in the context of disease. And it all pretty much starts from a plot that looks like that. And on the y-axis here, you can put your favorite attribute about yourself, be that health, resilience, strength, muscle, fitness, eyesight, whatever, and what you will soon realize that it peaks very early on or very early on in life, but then there's a progressive deterioration of that. And ideally, what we would like to do is try to extend that health span and try to understand the mechanisms that will allow us to live healthier for longer, not necessarily increase our lifespan, but certainly make us live a healthier life. And that is because aging is actually one of the top predictors for disease, as you can see in this plot, and that includes all common diseases, including stroke, coronary artery disease, type 2 diabetes, cancer, so on and so forth. So in my group, over the last few years, we have focused a lot on understanding how our cells respond to damage and how they repair that. And essentially, we're trying to understand this very fine balance. And genetic risk factors and environmental factors can actually affect that balance. And while our cells are pretty good at repairing a lot of the damage that they get, some of these unrepaired damage accumulates, and then at the cellular level, this can lead to apoptosis, which is cell death, or senescence, which is a state of permanent cell cycle arrest. So the cells don't die, but they go into this state of not dividing anymore, not proliferating, and instead they have a signaling role. And I'm going to talk quite a bit about senescence towards the end of my talk. And the senescent cells secrete a lot of cytokines and interleukins, and they have mostly they contribute mostly on inflammation, and apoptosis and senescence can lead to loss of regenerative capacity and eventually functional decline and aging. And on the other hand, somatic oncogenic mutations can lead to clonal expansion and eventually cancer. And not only this is a fine balance, but our cells' ability to resolve stress actually deteriorates also with aging, and that's pretty much the definition of frailty. And our cells are becoming less and less capable of dealing with the stress. So in my group, we're trying to understand these mechanisms and try to understand this high-dimensional regulation of its expression as a key to discover the mechanistic base of the physiology and disease of aging. And we use some wet lab components, but we're mostly also on the computational side. But I'm going to try to keep, for the purpose of this talk, I'm going to try to keep it as much as possible closer to biology. And we use long-read RNA sequencing for our work. We couple that with single-cell and spatial RNA-seq and a lot of data science and machine learning. And the goal would be to try to use all of these things to understand basic biological mechanisms. So our interest started actually a few years back, where us and others have found that RNAs actually can be co-translationally degraded. So as RNA is being translated, it can also be degraded. But the most important aspect of this work, that in my opinion, is that these sites where these cuts are happening on the RNA co-translationally are actually very, very highly conserved. They're very conserved across all vertebrates and much more conserved than surrounding areas, which indicates for someone who is doing a lot of data science, whenever we see conservation, we pretty much, that's almost equivalent to these guys also being probably very heavily regulated. So these cuts are not happening just randomly on an RNA, but they probably happen at very specific positions. Now, one of the major problems that we had back then is that we were heavily relying on short-read RNA sequencing, and that meant we had to artificially fragment the RNA into smaller pieces that we would sequence with Illumina sequence. And obviously, that's not ideal if you want to understand how decay of RNA happens, both at the 5' end and the 3' end. So we very quickly realized that we need to switch into something that would allow us to sequence full-length RNA molecules. And at that time, we decided we would go with nanopore sequencing. And I'm not going to go into much detail about how this works. But the important thing is that you can think of the RNA essentially translocating through a nanopore. And as different bases pass through the pore, essentially there are fluctuations in the electrical current that the device measures. And then you get this squiggle of an electrical current right here, which then is decoded into the bases, and that's how sequencing works. And the benefit of that is that you can sequence pretty much entire molecules. So I'm not going to go into details on what we did to ensure that we are capturing full-length molecules, and that involved also ligating adaptor at the 5' end of the molecules, because sequencing actually proceeds from the poly A tail towards the 5' end. So we want to make sure that we reach all the way to the 5' end. That will get you straight to the to some of the results that we got, and this is just one isoform, every individual thin line that you see here is one of the reads that we got. And I think, and the green light here indicates where the 5' end of the read was on this isoform, and where the 3' end of the read on this isoform was. And this is a quite extreme example, but what you can notice is that the 3' ends are very well defined, and most of the times they're at the end of the RNA molecule, which makes a lot of sense. But the 5' ends are much more heterogeneous. And not only you can find them along the entire RNA body, but also, as I told you before, you see these very prominent peaks here, which indicates that there's something about those areas that might be responsible for triggering decay. And actually, I'm not showing this data here, but at steady state, in HeLa cells, for example, we found that less than 50% of RNAs in total are actually intact. Most of the RNAs are actually fragmented either from the 5' end or from the 3' end. So if that happens in steady state, then we wonder what would be happening in cells that are not in steady state conditions. So this work was led by Sokat and Sulo, two postdoctoral fellows in my group. And they started out with a very, very simple experiment. They got HeLa cells. One they treated with DMSO, which is our control, and then the others they treated with different stressors. And then they performed long-read RNA sequencing with a 5' and adaptor ligation. I'm not going to go too much into it. And then they performed the nanopore sequencing with the idea that we would capture the RNA state, and then we would be able to compare normal cells versus stress cells to see what happens in non-steady state conditions. And to our surprise, what we immediately saw, and again, I'm showing you up here just a genome browser screenshot of a gene, which is the ACC3 gene. It's one of our top hits, essentially, where you can see that these are all the individual reads that we got, that upon arsenide treatment, upon stress, we have much shorter RNAs being sequenced. And this is actually true not only for just one gene, but for the majority of the genes, even those that have the adapters that we are very certain they are in our internal controls that we are sequencing all the way to the 5' end. And very importantly, there doesn't seem to be any specificity to this degradation mechanism. Instead, we also see that this equally affects downregulated genes, where you can see this off the diagonal length. So most of the isoforms are off the diagonal towards longer molecules in the control. In the unchanged, those that don't change their expression, and in the upregulated. So it doesn't really affect only one class of RNAs, but instead it seems to be applying across the entire transcriptome. Now, under canonical conditions, canonical mRNA decay, the prevailing view of how that works is that there are at least 3 steps. So the first step is the removal of the poly A tail, which is considered the rate-limiting step here. Then there is decapping, which is the removal of the cap of the RNA. And then there's exonucleolytic fragmentation of the RNA, both from the 5' end via XRN1 and from the 3' end via the exosome. So we want to see how much of that is true during stress-induced decay. So first, we knocked down XRN1 with silencing RNAs, and we immediately saw that we have a very clear increase in the length, and we fully rescue pretty much the length of the RNA, indicating that indeed whatever we're saying, the stress-induced decay is mediated by the exonuclease XRN1, and XRN1 plays a huge role in it. However, when we actually knock down DCP2, which is the decapping enzyme, or NOT8, one of the central components of the CCR4 NOT complex, we actually saw absolutely no difference. We saw that the lines were mostly overlapping between control and arsenite treatment in these two knockdowns compared to GFP, indicating that neither of the two, neither de-adenoylation, full removal of the poly-A tail, nor decapping are actually required for stress-induced RNA decay. So that was unexpected, and we also wanted to understand what might be happening, how this is organized. What are the logistics of these RNA decay essentially happening in cells? And in stress, essentially what happens is that first there is the when cells are stressed, there is the creation of these of stress granules, which are membraneless organelles consisting of RNP complexes, ribonucleoprotein complexes, and that happens because a massive amount of RNAs are exiting the translational pool because there is translation arrest during stress, and these RNAs essentially aggregate into these granules. And one of the first observations that we had is we got some data from early on from Roy Parker's group, and we overlapped what we found as the most stress-induced degraded RNAs with the stress granule transcriptome provided, identified by Roy Parker's group. And we saw a very clear enrichment of the top degraded RNAs in the stress granule transcriptome. So that gave us the idea that maybe stress granules are actually absolutely required for it. So we turned into U2OS cells, and we got the cells from Paul Anderson's lab, which they have a double knockout of G3BP1 and 2. And in these cells, the stress granules cannot be formed. So we compared that against wild-type U2OS cells. And what we find is that when we stress them with arsenite, in the wild type, we get very clear shortening of the RNAs. But in the absence of stress granules, pretty much there was no stress-induced RNA decay. And that was strong evidence for us that the stress granules are actually indispensable for stress-induced RNA decay. But besides that, we also wanted to understand what is the fate of these RNAs. Okay, we're starting to see them being degraded at the 5', but what is happening to them? And for that, we started using metabolic labeling experiments, where essentially, just in very broad strokes, you treat these cells with a nucleoside analog. In our case, that would be 5-EU, 5-ethylene uridine. And the idea is that during the metabolic labeling experiment, all the newly synthesized RNAs will incorporate at least one 5-EU in the RNA, whereas the pre-existing RNAs would have no 5-EUs. And we're doing direct RNA sequencing. So the idea was, if a molecule actually incorporates a 5-EU, would we be able to identify that directly from the nanopore sequencing if the 5-EU was leaving a unique mark in the electrical signal that it was unique from a normal uridine? So to do that, we had to do a lot of machine learning, and that we spent a lot of time on that project. And that work was led by Vlasta and Jessica in my group, and in collaboration with the Alexiou lab in the Czech Republic. And essentially, what Vlasta and Jessica wanted to do is they wanted to have a tool that starts from raw signal, could classify metabolic labeled RNAs based on 5-EU, had absolutely no dependencies on other software so that it's future-proof, and could also generalize to other datasets and not just our dataset. So they developed RNA-Kinet, and I'm not going to go into details here. There are very technical computational stuff going on here, which I'm not going to go through it. But just for those that are interested, essentially RNA-Kinet is a convolutional neural network as a first step to identify patterns in the electrical signal. And then it's coupled with a recurrent neural network that it then integrates these patterns across the entire read in order to make a final inference whether a read is 5-EU modified or not. And then we did many different tests for that, but one of the most interesting tests that we did is we treated cells for 2 hours with 5-EU, and then we quantified half-lives of RNAs with RNA-Kinet. And we went back and we tested against a completely independent data set that was produced from Bartel's Bartel group in a molecular cell work a couple of years back. And we actually found pretty good correlation with the half-lives calculated there. But not only that, we found that we had very good overlap between different replicates that we did, and also the more reads an gene had, the better our inference would be, which is a pretty good thing to see. As the sequencing gets deeper and deeper, the performance of the tool will keep improving. So we use that tool now to try to understand what was going on to these RNAs that were found as degraded during stress. So our experiment is we treat for 5EU, we label for 5 cells for 2 hours with 5EU. 1 hour out of that, we actually stress the cells. And then we want to see what happens during the recovery period to these, specifically to these RNAs that we were finding as the most degraded during stress. We run this through RNA kinetics, so we separate labeled from unlabeled RNAs. And essentially, what we see is that the RNAs that were the most degraded during stress were the ones that actually had the lowest percentage change in modified molecules. Essentially, that means that they had the less amount of modified RNAs of 90 minutes, indicating that they're progressively being degraded, fully degraded, and then essentially resynthesized. And there's essentially new synthesis without 5EU in this case, because we have stopped at 60 minutes, the 5EU. So that fitted very well with what we were predicting that was going to happen, which is once these RNAs are starting to be degraded to the 5' end, then essentially they have no fate, they have no other function in the cell, so eventually they're going to be fully degraded during stress recovery. So that leads us to this model where under normal conditions, the RNAs are happily translated by ribosomes, and that's blocking XRN1. But once stress hits, we have the formation of stress granules. And then for molecules that still have their poly-A tail and still have their 5' cap, there must be an endonucleolytic cap that creates an initial cap, which is then latched on by XRN1, which fully processes and degrades the RNA. And then eventually these RNAs get eliminated from the cell during cell recovery after stress. So that brings me to a second story, which also has to do with a stress response, but in this case, I'm talking about senescence. And senescence is a stress response to genotoxic damage, to DNA damage, and it acts as an antagonist to cellular health, especially during aging, because we have accumulation of senescence, global accumulation of senescence, pretty much all the organs during aging. And these cells don't die. They instead go into this permanent cell cycle arrest, but they're not also translationally inactive. They actually have a very potent translational program, which is called the senescence-associated secretory phenotype. It consists of interleukins and cytokines, and essentially senescent cells have a signaling role. And that, during aging, can lead to secondary senescence, which are some of the deleterious effects of it. It leads to increase of inflammation, which we don't always want, and sometimes counterintuitively can lead to tumor genesis. So we want to understand a little bit more about the senescence as a stress response. So this work is led by Matthew Pei in my group in collaboration with the Gorospi lab. And essentially, our story started with a proteomics study of senescent cells. And what we found is that the majority of, in this proteomic study, the majority of the targets that we were getting were actually endoplasmic reticulum stress markers. And that indicated to us that senescence as a stress response actually has basal increase in ER stress. And as you probably know, when you have ER stress, you need to activate the integrated stress response to essentially resolve that stress. And the integrated stress response can essentially is a pathway that responds to different stressors, including viral infection, proteotoxic stress, including ER stress, starvation, and others. But all of them actually converge into a single point, and that is that the phosphorylation of initiation factor 2 alpha, which essentially the goal would be to arrest translation initiation and shut down translation. Except that globally happens, except the transcription factor ATF4, which needs to be activated during the integrated stress response. And the way this is happening is that ATF4 is regulated by upstream open reading frames. And under normal conditions, ribosomes mostly cycle on these upstream open reading frames because they have enough ternary complex, enough non-phosphorylated EIF to alpha to essentially keep translating the upstream morph. But in the case of EIF to alpha phosphorylation, when you have stress, this becomes limited, and then some of these ribosomes will scan through and actually go and translate the main ATF4 mRNA. So if we look at this graph, essentially what we're saying is that the longer it takes for a ribosome to find a non-phosphorylated EIF to alpha molecule, the greater the chance that URF bypass is going to occur on ATF4. So you can think of it like that, in a graph like that. So you have the ribosome demand, and ternary complex availability goes down progressively because you get more and more phosphorylation. And at some point, you hit that sweet spot that activates the expression of ATF4, and you have activation of the downstream integrated stress response pathway. So as I told you, in senescence, we had this basal ER stress. And so we want to study, okay, we must have activation of the integrated stress response then. And indeed, we have very potent, when we looked in senescent cells, we had very, very potent phosphorylation of EIF to alpha. Also, we knocked down PERK, which is the kinase that is responsible for phosphorylation of EIF to alpha during proteotoxic stress, which is the ER stress. And indeed, we see that when we knock down PERK, we have a reduction of EIF to alpha P, indicating that indeed senescent cells have ER stress. But what we didn't expect was this. We had absolutely no expression of the transcription factor ATF4. In contrast to our positive control, which is thapsigargin-treated cycling cells to induce ER stress. So we really wonder why this might be happening. Why don't we get ATF4? So initially, we thought it must be we don't have enough mRNA. So we essentially transfected wild-type ATF4 in the cells to try to boost the expression of the availability of ATF4 mRNA. But this did absolutely nothing for senescent cells, whereas in cycling cells, we did see some expression. So senescent cells did not express ATF4, although we had it in abundance. So we thought that maybe that has something to do with the upstream morph bypassing mechanism. So we mutated the upstream morphs, and we changed the start codons of the upstream morphs into ATT, essentially deleting them. And when we do that, we do actually get quite potent expression of ATF4. So that led us to believe that, something is going on with the upstream open reading frame bypassing mechanism on ATF4. So for that, to gain more mechanistic insight into that, we performed ribosome footprinting experiments. I'm not going to go. I think everyone in the audience here probably knows what ribosome footprinting is. And essentially, when we do ribosome footprinting, what we do see is that in normal cycling cells, we do see this very potent block of ribosomes in the upstream open reading frame. In our positive control cycling cells treated with thapsigargin, we see this block is completely gone, and then we have expression of ATF4. And when we go into senescence, two different models of senescence, we essentially see the same thing as we see in cycling. We still see this block in the upstream open reading frame, indicating that this bypassing mechanism does not work in senescence. So that was very surprising to us in the beginning. We couldn't understand why. But then what we noticed is that consistently in all our experiments, we were getting substantially fewer ribosome profiling footprints for senescent cells, both models compared to cycling cells. Actually, when we normalized them and tried to plot ribosec over RNA-seq, we consistently saw we were getting much fewer ribosomes per unit of RNA in senescent cells. We did a lot of experiments to try to prove that. I'm not going to go into details here, but essentially, our working model at that point was that we were focusing on this sweet spot right here, assuming this flat line here, that's the demand from ribosomes would be relatively constant. But actually what happens is that in senescence, you have much fewer ribosomes actually requesting the ternary complex. And that means that this sweet spot for activation of ISR would be much higher. So essentially what we need is much higher phosphorylation of VIF to alpha in order to reach that same spot. And that essentially means that senescent cells would be desensitized to stress. They wouldn't be able to respond to stress at all. So we tested that. We additionally stressed our senescent cells with tapsigargine to introduce additional proteotoxic stress. We also did that with amino acid starvation, and we did so that we need to have very high expression of VIF to alpha phosphorylation in order to get any expression of ATF4, even minimal. And that also translates in downstream targets of the ISR and the UPR, where we see that we don't have activation of the downstream programs for activation of the stress response. And not only that, but this unresolved stress actually remodels the senescence-associated secretory phenotype, sometimes making it worse and making some of the factors being more expressed and some others less expressed. But generally, there's global remodeling of the senescence-associated secretory phenotype, which we can actually rescue if we can introduce back our ATF4 delta construct, where these upstream morphs can be deleted and essentially ameliorate this observation. So this is our working model, and I'm not going to go through much because I already explained most of these things. But essentially, the bottom line is that in senescence, when you have stress, you need very high expression of VIF to alpha P, because senescent cells are globally desensitized for the stress response, and that leads to stress-induced secretory remodeling. So with that, I would like to thank my team. I think I mentioned everyone during the presentation: Sokat, Matt, Cedric, Chris, everyone, and all my collaborators, particularly the group of Miriam Gorospi. And with that, I would like to thank you and happy to take any questions if there's time.
Thank you, Manolis. That was a beautiful talk. I want to remind everyone that if you have questions, please write them in the Q&A box, and I will relay them to Manolis. You can answer them here live, or you can send them an email asking if we didn't have time to ask. So while we wait to see if you guys want to ask him questions live, I have a question for you, Manolis. It's a little bit of a technical question. I'm very fascinated about your end-to-end sequencing of RNA molecules. How many of the reads are actually give you end-to-end information? Because as far as I know, Nanopore, you have quite a few molecules that have incomplete sequencing, right?
Yes, that's a very interesting point. So that's precisely the reason why we went there out of ligating adapters of the 5' end, because we wanted to have in all our libraries have an internal control where we know that if we actually find that sequence in our reads, we know that we reached all the way to the 5' end. So for relatively shorter molecules, up to maybe 3 kb, I would say you can get very good resolution in the sequencing. If you start going much longer than that, then it becomes a little bit more tricky to get full-length molecules. But for the majority of them, when we look at the distribution of read lengths, let's say with 5' end adapters and without 5' adapters, we still see pretty much the same distribution. So I would say it's quite a close representation of what you actually have in the cells. There's certainly some, and you need to be careful. I think if you want to do these studies, it's worth having these adapters ligating for the peace of mind of sequencing all the way to 5' end. But generally, I think it is quite accurate, especially for genes up to 3 kb.
Okay. Wilfred Giblett has a question for you. He says, great talk. Does the degradation of RNA affect gene expression profiles? Would Nanopore be better than Illumina for accurate counting?
Yeah, that's a big question. Personally, yes. I think it would be more accurate, especially if you have one of the bigger PromethION devices that you get lots of reads. I think it's actually the best because you don't have any PCR amplification in direct RNA-seq. You don't have any reverse transcription requirements. So you really sequence what's there. So I would say it's more accurate, but you also get a lot of, substantially more information. You get all the splicing information. You really know where the 5' ends of your molecules are. You can quantify how many full-length molecules you have. So I would say, in most respects, I think it's a very good assay for a very informative assay. Yes. I don't know if I fully answered the question. And RNA degradation, yes, it affects abundance, but after the stress discovery, at least in the experiments that we've done so far. So we don't see the levels of RNA changing that much. So degradation starts at the 5' end, but these do not get immediately eliminated from the cell. Somehow, it takes that time. It's a little bit difficult to know exactly the timing because you have to do, very fine time scale experiments to really figure that out. But I think most of the abundance is going to be after the stress gets resolved.
I think Wilfred said that you answered perfectly. I have one more question for you, which is for the people in the audience that are dying to use your strategies in their own work. Do you have tools for the analysis of the data available?
Yes. Well, RNA-Kinet is fully open source if you want to use it, and it's available, and we're happy for people to use it and send us feedback if they encounter any problems. We're happy and very active to resolve them. And most of the other tools, we're happy to guide you. We have most of the tools available on GitHub. So, if you can't find something, just shoot me an email. I would be very happy to help and, guide you how to use the tools. Nanoplane is also available to quantify differential expression analysis, differential length analysis. So yes, just shoot me a message if you want to use any of these, and I'll be happy to guide you.
Perfect. With that, I think we reached the end of our time. Let me thank you both again on behalf of everyone that's participating for 2 beautiful talks. And again, if people in the audience are thinking about questions that they forgot to ask, both Manolis and Thomas will be happy to receive an email from you and continue the discussion offline. So with that, thank you all for joining, and we will see you in the next RNA Collaborative seminar.
Thank you. Bye.
Bye-bye.